directed to the email that you used for registration. Please feel free to use the chat box feature to send us questions. Questions will be answered to the best of our ability at the end of the webinar during the Q&A session if time permits. If we do not have time to get to your question, we will compile and consolidate the questions and do our best to provide answers to you via the email that you provided for registration. My name is Kayla Cantrell. I am a study director and toxicologist at the Institute for In Vitro Sciences, and it is a pleasure to be your moderator today. Thank you all for joining us for the first of 2 webinars on the derivation of a point of departure from in vitro methods for the use of skin sensitization risk assessment. We are very excited about this approach and its applicability to risk assessment as proposed by Dr. Andreas Natsch and his team. IIVS has been able to work with Dr. Natsch and offer this novel methodology as of last year, as well as offer all of the in vitro and in chemico methods that we discussed today. Our first webinar today will involve an introduction to the in vitro and in chemico skin sensitization methods involved in calculation of a point of departure by Dr. Argel Islas-Robles from the Institute for In Vitro Sciences, followed by the incorporation of these methodologies in deriving a point of departure presented by Dr. Andreas Natsch from Givaudan. Without further ado, allow me to introduce Dr. Argel Islas-Robles. Dr. Islas-Robles has a bachelor's degree in biopharmaceutical chemistry from the University of Guanajuato, Mexico. In 2019, he obtained his PhD in pharmacology and toxicology from the University of Arizona. In the same year, he joined IIVS as a toxicologist and study director overseeing assays for the assessment of ocular and dermal toxicology. In the last couple of years, he is focused on skin sensitization approaches and currently is our program leader for skin sensitization at IIVS. Additionally, he works with government, industry, and other nonprofit organizations for the validation, implementation, and promotion of novel non-animal strategies in toxicology. Dr. Islas-Robles, thank you for joining us today. I will turn the floor over to you.
Thanks, Kayla, for the introduction. Just give me 1 second to put my slides. Okay, let me know if you can see them already. Okay, so thank you everybody for joining the webinar. Today I'm going to do a very brief overview of the skin sensitization methods as an introduction for the calculation of the points of departure that we are going to talk about today. I'm also going to stop sharing my camera just to don't spend a lot of internet band. So first of all, I'm going to start with a very brief outline of my short presentation. As I was mentioning, I'm going to do an overview of the skin sensitization methods and also the mechanism leading to skin sensitization. We are going to talk about those OECD methods that are used for the calculation of the point of departure, and also some of the defined approach that are used to establish the sensitization potential. And finally, I'm going to give a very brief introduction of the development of the method that we are going to talk about today for calculating the point of departure from the sensitization in vitro data. So as we all are aware, and we are here, is because the skin sensitization that typically is what we call allergic contact dermatitis. This is typically characterized by redness, skin rash, and urticaria, and is divided in 2 different stages. One is the initial exposure to the test chemical or induction phase. In this part, there is no symptoms of allergy and is where the foreign molecule enters to our body, and is also when the sensitization phase occurs. When there is a repeated exposure of the test substance is when we are going to develop the symptoms of allergy, and this is known as the elicitation phase. So just as a reminder for everybody, this is the mechanistic overview of how skin sensitization occurs. And as we know, the allergens, the first step is to enter through the skin. Then they bind to proteins for a process called haptenation. And then these complexes interact with different cells in the skin, leading to its activation, for example, dendritic cells and keratinocytes. In this figure, I'm showing the maturation of the dendritic cells, which migrate to the lymph node. And in this part, they interact with the T cells for presenting the antigens. And when the T cells gets activated, they travel to all the body. During a new exposure to the test chemicals, the T cells are going to recognize the allergen, and this is going to lead to an inflammation response with the activation of the different cells, keratinocytes, dendritic cells, chemokines, cytokines, and you are going to see the symptoms. Again, this is the sensitization phase and the elicitation phase. For the assays that we are going to talk about today for the calculation of the point of departure, we are looking at the haptenation process in the KDPRA assay, also the activation of keratinocytes using the keratinosense, and also the maturation of dendritic cells using the HCLAT. In this figure, I just wanted to show very briefly the adverse outcome pathway for skin sensitization. This just summarizes those events that are needed to happen for a test chemical to be considered a sensitizer. And we have from the organ response that typically is assessed in vivo or also in human. Today, we are going to talk about the local lymph node assay or LLNA, as well as here are included all those in vitro assays, in chemical assays, and in silico assays that are helpful to predict the sensitization potential. I should mention that these methods that are included in this slide are already validated and adopted by OECD. So starting with the first assay is the kinetic direct peptide reactivity assay or KDPRA. This is outlined on the OECD testing guideline 442C. And we are looking here at the endpoint of haptenation, which again is the covalent binding of the different substances or haptens to skin proteins. In the assay, what we are doing first is diluting the test article at different concentrations to have it react with a peptide that contains cysteine moieties. What we are trying to see here is the binding of the hapten to the cysteine peptide, and then we add a fluorescent dye known as monobromobimane or MBRB. And these compound pretty much interact with the free cysteine moieties to give us this compound, which actually have some fluorescence that is able to be detected in a fluorescent parade reader. And then we calculate the percentage depletion in reference to the solvent control. So one important part of this assay is that it generates kinetic data, given that we test these different time points from 10 minutes to 24 hours and also at different concentrations ranging from 0.31 to 5 millimolar. The data is then plotted as the inverse logarithmic scale versus the concentration at each of the time points that are tested. And the main point for this is to calculate the reaction rate between the test chemical and the peptide. And what we wanted to obtain is the reaction rate again, that is known as the log Kmax. And we can use this value to predict two different categories of potency in the sensitization, which is those strong sensitizers or category 1A versus those that are not strong sensitizer or category 1B or not categorized. It's important also to mention that the KDPRA was one of the first validated assays that was allowed to alone predict sensitization potency. And it also has been shown that the rate constant, the log Kmax, is one of the strongest determinant of potency among the parameters of all the other validated methods. The other method that we are going to talk about today is keratinosense. And in this assay, what we are looking at is the keratinocyte activation pretty much by the induction of the antioxidant electrophile response element. And this set of genes is typically controlled by the promoter of NRF2. And when this construct is induced, we are going to see a luciferase gene activity that is able to be quantified. Typically, NRF2 is associated with the repressor protein KEAP1, and when there is an hapten present, is going to interact with KEAP1 and then is going to release NRF2 to produce this gene expression. So a very brief outline of the assay. We have 12 different concentration of the test samples that are dosed to keratinosense cells, which have this construct already. These are treated for 48 hours. Then we proceed to lysis the cells and then add the luciferase substrate to read it in a luminescence detector. I also should mention that in parallel, the cytotoxicity is evaluated using MTT. This is the typical data that we obtain in the keratinosense. In this specific case, we are looking at the strong sensitizer DNCB, and in the left side, we are seeing at the fold induction. Pretty much this is the expression of the genes in comparisons to the solvent control. In the right hand, we are looking at the viability, and on the x-axis, we are looking at the concentrations that are tested. So in this assay, we have some parameters that evaluate the induction. For example, the Imax, which corresponds to the maximum induction observed. Also the EC 1.5, which is that concentrations that leads to fold induction above or at 1.5 folds. And also we have those parameters that evaluate viability as the IC30, which is that concentration that leads to a reduce in 30% of viability. Similarly, we have the IC50. And using these parameters, we are able to predict if a test chemical is going to be negative for sensitization or positive for sensitization. I also wanted to point out that IIVS participated in the validation of this method, and here is the reference also for you to have. And additionally, there is another parameter that is taken into account for the calculation of the point of departure, which is the EC3 values. This is the concentration that leads to a threefold induction versus the solvent control. For most of the acid, I'm going to mention these parameters given that they are used for the calculations that you are going to see in the next presentation for today. Moving to the last method that I'm going to talk about today, this is the human cell line activation test, or HCLAT. And what we are trying to see in this assay is the activation of dendritic cells, which is the third key event on the outcome pathway for skin sensitization. When we have the dendritic cells and they maturate in we can see changes in the surface markers, also in the chemokines and cytokines that they release, and also in the genomic transcripts. For the HCLAT particularly, what we are looking is the expression of specific surface markers, which are CD86 and CD54. For the brief outline of the assay, we are having here THP1 cells, which are treated with 8 different concentrations of the test article or test substance for 24 hours. Then we proceed to harvest the cells, and then we stain them with antibodies that specifically recognize CD86 and CD54. Then this is analyzed via flow cytometry, and in parallel, the cytotoxicity is analyzed using the propylium iodide staining. For the prediction model, the parameter that is calculated is the relative fluorescence intensity using this formula. And we evaluate the RFIs for CD54 and CD86. For CD54, if they are higher than 200% compared to the controls, we are going to have a positive classification in the sensitization potential. And for CD86, if the RFIs at any concentration are higher than 150%, we are also going to consider this result as positive for sensitization. I also wanted to include other parameters that again are used for the calculation of the point of departure. One is the CV75, which is the estimated concentration that is required to elix 70% of the cell viability. This is typically calculated in the dose range finding assay for the HCLAT. Other parameters are the EC150 and the EC200. These are the effective concentration that lead to an RFI of 150% for CD86 or 200% for CD54. One other important value is the minimal induction threshold, or MIT, and this is the minimum of the EC150 or the EC200 that is calculated for the test chemical. Moving on, as I have mentioned for all the other methods, typically we have a binary classification in where the test samples are classified versus positive or negative. However, these methods are not an standalone to classify the sensitization potential. So there has been different approaches that are utilized in parallel to allow the final classification. One of the methods that is outlined in the OECD guideline for 97 is the 2 out of 3, which uses the data of DPRA, H-clad, and keratinosense. In this case, note that this is not the kinetic DPRA, this is the standard DPRA, in where pretty much we are expecting to have at least two concordant results to do the final prediction. One strategy is to start with two of these assays, and if they are conclusive, we end up having a negative or positive, and if they are not conclusive, we will perform a third assay. This strategy has been shown to be pretty effective in comparison to human data. Here I am showing the sensitivity, specificity, and accuracy of the 2 out of 3 approach. The OECD guideline for 97 also have some integrated testing strategies, including in silico methods. This is one of the integrated strategies. This is version number 1, in where the data obtained from in silico, particularly from Derek and the OECD toolbox, are scored, same as the data obtained from the DPRA and the HCLAD. And based on the scores that are added, given the data that is obtained, we are able to classify the potency of the test sample into GHS classification 1A, 1B, or not classified. And please note that this is just classification according to the GHS. We don't have very continuous classification of the potency. In this little table, I'm just showing, the categorization and the overall prediction of this endpoint. Moving forward to the introduction of the topic that we are here today, is about the prediction of the LLNA EC3 values using non-animal methods. Back in 2015, the group of Andreas uses the keratinosense data plus the kinetic peptide binding data. And I wanted to point out here that this is not the standard KDPRA. The work that was done in 2015 was using a different peptide, but pretty much this assay is the predecessor of KDPRA. And then using this data, different regression analyses were done against the LLNA EC3. And in my next slide, I'm going to explain a little bit about this value. But anyway, using the regression, we said, well, Andrea selected this model that have the most powerful predictive constant. For example, in this graph, we are showing here the predicted EC3 values versus the EC3 values that are on the database from the chemicals. The black line corresponds to the prediction, while the dotted line corresponds to the actual values. In this regression model, what was used was the log Emax from the kinetic peptide binding and the EC 1.5 and EC 50 from keratinosense, as well as the vapor pressure. And it was shown that this type of calculation was useful to provide a point of departure for further potency assessment and risk assessment. So talking about the LLNA EC3 values, first to define this, I have this little paragraph that I take for this reference. And the EC3 is pretty much a dose response is obtained from the dose response from the LLNA assay. And this is the dose that gives us a stimulation index of 3.0. In this table that I have here, I just have one example of the classifications, potency classifications that can be assigned based on the EC3 values. However, besides the classification, one important aspect is that this generates a continuous potency value that can be used in risk assessment. And I just wanted to show one of the strategies that we have been used at IIVS, that is first determine the hazard classification of the test compound using the 2 out of 3 approach. Following this testing, if we have a negative, we will be done and predict the test chemical just as not sensitizer. However, if we end up having a positive classification, then we proceed to perform the KDPRA to calculate the predicted EC3 values, which can then be used further in risk assessment. So with that, I wanted to end the introduction for the topic that we are going to discuss today. And again, the questions can be asked in the chat box, and we are going to try to respond by the end of the presentation. Thank you.
Allow me to share my screen quickly. All right, you should be seeing a presentation. Thank you, Argel, for your presentation on current skin sensitization methodologies and defined approaches using non-animal methods. It's incredibly important that all parties have insight into these assays, as it provides a foundation of understanding why these methods are so important for the calculation of a point of departure for skin sensitization. At this time, I will introduce you all to Dr. Andreas Natsch, who will guide you through the second half of our webinar. Andreas Natsch studied at the Swiss Federal Institute of Technology from 1988 to 1996 and was awarded his PhD in environmental microbiology. After his postdoctoral studies in molecular biology at the Centro Nacional de Biotecnológica in Madrid in 1997, he moved to the research department of Givaudan in 1998. Here, he elucidated the biochemical mechanisms of body odor formation in order to design new deodorant ingredients. Since 2008, the research focus has shifted to the development of in vitro assays to study toxicological risks without the use of animal testing. This work focused first on the mechanism and the kinetics of reactivity of skin allergens with peptides, and then led to the development of the keratinescence assay to study allergens, which was globally adopted in 2015 by the OECD as the first biological animal-free assay to screen for skin allergens. In 2021, the 2 out of 3 testing approach and the kinetic DPRA, developed together with BASF, were adopted by the OECD. Parallel work of his lab led to 3 OECD guidelines in the ecotoxicology field to replace acute fish toxicity test and bioaccumulation test. In his current position as Senior Research Fellow, he leads the in vitro molecular screening group at Givaudan to evaluate risks and bioactivity of novel ingredients early in the development pipeline. Dr. Natsch, thank you for joining us today. I'll turn the floor over to you.
Hello, everyone. Thanks very much to the Institute of In Vitro Sciences to invite me to give this webinar. And thanks very much for all the introduction, which helps us to get ready for the subject. So here we are with the technology.
Perfect. It looks great, Andreas.
So I will, in this first part today, first give you an overall approach how we do this potency assessment, quickly go through the input data and the databases and how we utilize them to build these regression models. I will show you how this can very easily be applied in practice with a spreadsheet which we provided, and then deep dive into the predictivity, what we can predict and how accurate we are. This will lead me to discuss specifically why we have under and over predictions, and show you some data on how robust these models are, and also about data redundancy, which will be quite important also for what we will discuss next week. I will also take this opportunity to give you a brief update on the SOP for keratinosens and KDPRA, which we updated last year. So when we talk about risk assessment without animal testing, what we need is really a quantitative measure of the risk. And our overall approach here is that we combine the continuous and quantitative data, which we have from keratinosens, age-class, with the kinetic DPR rate constants, and then use that for a multiple regression versus the LLNA data. This leads us not to the LLNA EC3 value, but to something which we can consider most likely LLNA EC3, as we can determine it from in vitro data, which we then can use as a point of departure for risk assessment. What is then needed is, of course, an uncertainty assessment. We have to check prediction accuracy, and we do that by looking at similar molecules where we have both in vitro and in vivo data. And this will help us to assess how accurate we are in a specific chemical domain. This part I will not cover in detail today, but you will find it in our 2018 publication. And finally, when we have done that, we can use these uncertainty factors to adjust this point of departure to then arrive at a value which we can directly use, for example, in a scheme like the quantitative risk assessment 2, which is widely used, especially by the fragrance industry, to determine safe use levels. Now, when we talk about quantitative data from individual tests, it's always important to keep in mind that all these tests initially were just validated with the binary prediction model. Is it positive or negative? And that was basically what the validation authorities looked at. But all these tests do, as Argel has shown you, continuous and quantitative dose response information, which was in most cases, except for the kinetic DPRA, not really part of the validation, but in the validation, we also generated this data in multiple labs. And just to briefly repeat, keratinosens, we have two doses, the EC 1.5 and EC3 for the luciferase induction, and we have the IC50 for the cytotoxicity. In the age-class, we have the EC 150 and EC 200, again, for induction of the cellular markers next to the cytotoxicity measure CV75. And on top, we have the MIT, and the MIT is just the minimal value of the EC 150 and EC 200. And it is actually the MIT which we are using for all the statistics I'm going to show you today. We, for all these chemicals, of course, have the classical DPRA, but this one we only use for classification for the yes/no answer. And then we have the kinetic DPRA, which gives us the rate constants. When we turn to the databases we have, then we see that for the LLNA, keratinosens, and kinetic DPRA, we have a quite large database, in total 203 chemicals, and 188 of those chemicals also have the age-class data. Then we have a data set with so-called curated LLNA data put together by the OECD, and these are 149 chemicals where we have also all the in vitro data. And we have to remember all the time that the skin sensitization field is quite unique in terms of the size of the data set for both in vitro and in vivo data. Now, to go into the statistics, we express all data in molar equivalents because some tests are normally done in a weight per volume mode, and others are done in a molar mode, but we express everything in molar equivalents. We log transform all the values, and then we do a linear transformation so that all data are normalized to 0 and expressed in a positive scale. These linearizations actually do not affect the outcome, but they will make the reading of the equations simpler and make everything a bit more transparent to understand how the different things add to each other. And then this gives the final input parameter, which are the KS log EC1.5 normalized and so forth. So it's always a logarithmic normalized value which we enter, and we also have logarithmic and normalized values for the animal data for the LNA. This data we then just entered into classical multiple regression, which are maybe the most similar simple models to combine multilinear data, and they just give us linear combinations of the input parameter to predict our target, which is the EC3 in the LNA. Now we have 3 assays, and actually we can do different models, and we did different models for partial and complete evidence. So we have 3 combinations where we just have 2 tests. We call them equation 1, 4, and 6, where we had either keratinescence or HGLAT combined with KDPRA, or the 2 cell-based assays combined to each other with each other in equation 6. And these 3 models can use if we have partial evidence, if you only have 2 tests, or if you only have 2 positive tests. And then finally, we can also combine all the evidence, and that is what we call equation 5. And how this looks like, very simple. We end up with an equation where we have the predicted EC3, which is combined from a constant, and then we have a coefficient for the reactivity. We have a coefficient for EC 1.5 and the cytotoxicity in keratinescence. And finally, we see that we have a reduction of the potency if a molecule is highly volatile, because it will evaporate quickly from the skin in the animal test. Very similar equation you see for equation 4. When we exchange the keratinescence with the HGLAT, we get a quite similar contribution of reactivity. And again, it's the cellular activation, which is here the MIT, and the cytotoxicity, which contribute to potency, again with a negative contribution of volatility. Now, as I said, these are quite simple models, but that's also the beauty of it, because it's really transparent. You see these equations, and in principle, with these equations from the previous slide, you will be directly able to calculate the PEC3. So there is no proprietary software or hidden algorithm which is needed here. But we wanted to make it even more easy, so we made a publicly available spreadsheet which you can download from the Altex web page, just together with our paper. And it will look like you see it here, and we quickly go into that. Just quickly to check, does everybody see the Excel sheet? Is that okay?
Yes.
Yeah, perfect. So there is a blank sheet, of course, that's what you use. You just have the yellow fields, and there are explanations here, but basically you just need to take from the different test reports the values, enter them for the 3 tests in the yellow fields, and then all the calculations are done automatically. And I have also in that, by default, 2 examples introduced, so where you can see how this works. So here we have cinnamic aldehyde, which is positive in the keratinescence with an EC 1.5 of 16 micromolar. It has a rate constant of significant reactivity in the kinetic DPRA, and it's positive in the HGLAT with an MIT of 10 microgram, which is transformed then into micromolar. And based on that, since we have all 3 tests, we will have the calculations of all the models. And what you see already here, and this will be a recurring theme here in this webinar, you see actually we calculate 4 times a very similar EC3 value. So this shows that with partial or complete evidence, we have quite a robust prediction that cinnamic aldehyde has an EC3 of around 1%. And that's pretty much close to what we see in vivo, as you will see later, for cinnamic aldehyde. And a higher potency is seen for DNCB, where we see, again, quite similar with 4 different models with partial or complete evidence. We see this EC3 predicted of around 0.2%. So we are back in the PowerPoint, Kayla, can you confirm?
Yes, looks great.
Yeah, very good. Okay, just to be sure. Okay, so that makes it quite easy for application in praxis. Once you have the in vitro data, you can just go on and do these calculations. Now, what is the predictivity uh, uh, of that model? And here is shown for the equation 5, where if you have all the information and you see the PC3 in vivo on the x-axis and the predicted PC3 on the y-axis. Now, of course, you see this is not a perfect fit, far from a perfect fit. But what we can say is that around 75% of the chemicals are within a margin of error of 5, and 75% chemicals are less than, and that is what's important for regulation, less than threefold underpredicted. Now, and what we have to acknowledge that this uncertainty also includes the variability of the LNA. All the LLNA values are, most of them come from one single in vivo study, which even is with pooled lymph nodes, so meaning that there is no real statistic behind it. So we have the variability of the in vivo data here, and of course the predictive limitations of the LNA. And thus it's important to also look what the LNA, the current gold standard animal method, looks like when we compare it to the human data. And that's what you see on the second graph on the right. Here we have on the x-axis the PEC3 in the LNA compared to the human data expressed as the DSA-04. And you see that this correlation is lower than the in vitro to in vivo correlation. So because also the LNA is not a perfect, or far from being a perfect mimic what happens in humans. But again, this is not just the guilt of the LNA, it's also the human data which have quite a high uncertainty below. So we looked at this predictivity also with some other assessments. One thing we did, we just looked at the fold misprediction for all the chemicals. How far off are we in the geometric mean or the median? And this is around factor of 3 off for the geometric mean and 2.5 for the median. And then we see how many percent of our chemicals are over 5 or 10 times under or over predicted. And for the over 5 times under predicted, we are at around 16 to 18%. And what is interesting again here, we have the two partial models, equation 1 and 4, where we combine keratinosens and KDPRA, or keratinosens and HGLAT. And the overall misprediction is quite similar to equation 5. This means having the third test doesn't really improve our prediction very much. Finally, we also looked at this predictivity for case studies, and we selected as case studies those chemicals where we have at least 5 independent LLNA studies in the OECD database. And these are around 20 chemicals listed here. The good thing is here we know that the final LLNA value from all the studies together has quite high certainty, but at the same time we also know the variability of the LNA for those specific chemicals. And what we see is that for those chemicals where we have quite a high certainty for the LNA, we also have a relatively good prediction of with our models. And again, you see that we have quite similar predictions with the 3 models with partial or complete evidence, which indicates that we have certain flexibility which model we want to apply. I zoom in some of the chemicals of that table here. So for example, you see in top aniline, which is a very weak sensitizer. In some LNAs, it's negative. In some, it's positive. We have 14 studies ranging from 13% to non-sensitizing, meaning over 100%. This chemical is predicted by all the models in the very weak range of 50%. And then as an interesting case, we have citral, which has a LNA of EC3 of 5.8, but it comes from 16 studies, which are quite all over the place from 1.5 to 27%. And we would predict with these 3 different models a value of 5 to 9%, which is very close to the weighted mean of 5.8%. There's also one chemical here, which I want to highlight, which is clearly worse, where we predict the weak sensitization potency which is tenfold under predicted. And this is a primary amine. And as I will discuss, and that is also shown in the OECD test guideline, this is actually a chemical which is a chemical class which is outside of the applicability domain of the kinetic DPRA. The reactivity of this molecule is not seen because you need a metabolic activation to the aldehyde to occur first that these chemicals can sensitize. And then below you see also strong sensitizers, isoeugenol and catone, where we see this strong sensitization potency predicted. Again, looking at uncertainty, we looked at the under predictions, and I cannot discuss all the single chemicals here in this webinar, of course. In the second paper, which we published, we have a supporting information where we discussed every single chemical for under and over predictions. Here, I just want to state some general observation which we find when we look at under predictions, meaning where we get the sensitization potency predicted too weak. So we have 6 chemicals which are clearly too weak. We find them as weak sensitizers, while there are moderate sensitizers in the LNA. And this can include primary amines, which are, as I said, prohaptens, which need metabolic activation, and some amine reactive chemicals, which are outside of the applicability domain of the kinetic DPRA. On the other hand, we also find 12 chemicals where we see that there are significant sensitizers. The prediction is between 0.05 to 5%, but they are even worse in the LNA. So they are strong to extreme sensitizers in the LNA. And as a general observation, we see that at the upper end of the scale, the dynamic range for the extreme sensitizers of the model is limited. We would see that these are chemicals which we need to be very careful with because they are significant sensitizers, but we are not absolutely accurate for the in the extreme sensitization range. However, such chemicals are hardly would ever be considered for inclusion in consumer products anyway. Then we also have over predictions, and here we have a few interesting observations. First of all, when we had these discussions at OECD about the 2 out of 3, the concern was always that we have false negatives in the 2 out of 3, but we have also cases where the 2 out of 3 is positive, but the LNA actually is negative. But when we look inside into these chemicals, 4 of these 6, we know that they are very important human sensitizers, or we see that they are really strongly alkylating chemicals, and these alkylating chemicals normally are always sensitizers. In this case, we don't know why the LNA was negative, but we would certainly predict them to be sensitizers. And the other 2 chemicals here, is propylparaben and benzocaine, which are known as sensitizers from the clinic, but they are quite weak, and we would over predict their potency. And then we have 5 very reactive chemicals, which we clearly over predict. They are quite volatile. And in our model, we correct for volatility, but obviously not sufficiently. These chemicals, these are especially small acrylates. They evaporate immediately from the LNA conditions, but they are not evaporating in the closed in vitro assay, of course, and we would over predict them, but we can also say this is a conservative assessment. If somebody is exposed to these chemicals under occluded conditions, then of course they would be relevant sensitizers, as is known for acrylates. And then finally, we have 5 chemicals which are over predicted, but here, and that's discussed in detail, they are really relevant human sensitizers which might be under predicted in the LNA again against clinical data. Another aspect is robustness of the model and redundancy. With robustness, I mean whether this model is highly affected by just the specific chemicals or the specific tests which we selected, or whether we have something which is quite independent of the exact chemicals we used. And when we talk about redundancy, the question is really how many tests do we need and how many more tests the OECD needs to validate to get a perfect prediction. And what I already mentioned, when we looked at the predictivity and these case studies, we find often very similar predictions with the three models, and the mispredictions are quite similar. So this can be further illustrated here, and I plotted here all the chemicals which have three positive in vitro tests. And on the x-axis, you see either the combination carotenogens and KDPRA, or HClad and KDPRA, and on the y-axis, when we use all the three tests. And again, what you see here for almost all chemicals, the correlation is very good, meaning that actually in all those cases where we have three positive tests, we could have stopped testing after two positive tests, and we would not come to a much different outcome. So partial evidence can be sufficient, and this will raise a question: will we find new OECD tests which will provide a non-redundant additional information? And I will come back to these questions here in the second webinar. So that's just a teaser that you join again next week. Finally, I mentioned we will do an uncertainty assessment. When we have our point of departure, we will look how certain are we in a specific chemical space. And when we are quite close to the prediction for very closely related chemicals, we would use a factor of 2. And if we have less certainty, we would use at least a factor of 3 additional uncertainty factor, which we wouldn't have in the core, where we would directly use the in vivo data. With this, we cover the 75th percentile of underprediction. Now, some people would say, well, in statistics, you always have to be sure you need a 95% percentile, but we have to keep in mind that we in toxicology never work with 95% percentile assumption, because uncertainty is also factored in the assessment factors, which we then have in core A, which can be up to 400 in the case of deodorants. So we don't have to have all the uncertainty at this level of the assessment. So finally, these two publications, they are open access. You can download them from the Altex webpage, and especially they come with a lot of supporting information where you find the full database on all the test results, linked also to all the results from the OECD database. So everything is ready also to further modeling and find further approaches. And this follows up from the two earlier publications, especially the 2018 publication in Tox Science gives a lot of case studies how we apply that in a risk assessment. The earlier model is a bit different because there's another peptide assay, as Argel has said, but the update which we have here now, fully based on the OECD methods, in principle can be applied for case studies in the very same way as we have done it before. Which brings me finally to the conclusions for our first part of the webinar. The key event-based test guidelines, all of them next to hazard ID, they deliver continuous quantitative data, and this quantitative data, they do contribute to potency predictions. That's very clear and clearly an added benefit of these tests. The regression models are just one possible way to integrate this data. There are a facile and transparent way to derive something which is a continuous point of departure. And that's different to many other models which had been presented in the literature where people just tried to predict classes of sensitizers. What is really special here that we want to work in a continuous scale to give a quantitative value. And what I've shown you here already is that we do have data redundancy. Two tests give often a very similar prediction as 3 tests, which is good because it shows we have robust tests and we already have most of the informations. But it could also indicate that we are in a as good as it can get situation for a global model, and we will see in the future whether it's possible to further improve it. But certainly, where we are now, we can apply it. We have this simple application using our spreadsheet. Now, when we talk in OECD terminology, what we have here is an in vitro only defined approach. Defined approach has been defined as something where you don't need any expert judgment. So where you have a, what is in OECD terms, a standard data interpretation procedure, meaning it's a clear algorithm where you don't need any judgments. So with this approach, we have a defined approach. Using this in vitro data, you directly arrive at your point of departure. And some of you who know the field well, they may have recognized that we did not include any in silico evaluations. Many proposals used in silico models as further inputs, so combining in silico evaluation with in vitro evaluation. And that is not the case here. That doesn't mean that we think that is useless or shouldn't be done, but these additional lines of evidence can always be used to refine the assessment. We can look at the data, we can look at the molecule, we can look at the structural alerts, we can look at the output of the in silico evaluation and see whether it aligns with what we find based on the experimental in vitro data. But because we don't add them in the model directly, they are not like used up already and would then be used in a double assessment if they would be consulted. So we can really consult those type of information sources in parallel to what we do here. One important point is maybe to data reproducibility and data quality, because as I said, historically these tests were developed for the yes/no answer, and then it doesn't too much matter how clean your dose response curve is as long as you're above or below the threshold. But of course, to get a good prediction with quantitative data, you need to get good in vitro data quality. And so you need good quality dose response curve. And what we see in an experienced lab, the repeatability of the dose response information is quite high. So here is just given the average and standard deviation for 618 runs, which we've done on the keratinosense assay. It's not, you see at the point at the right, you have a big standard deviation, but that's just the upper scale of what we do in the positive control run. But you see that at the EC 1.5, we have quite a low variability over 600 independent runs, which also indicates that the keratinosense cell line is very stable over the years. So we don't have a shift of the data over the years. And very similar also for the kinetic DPRA, when we look at runs over time, that's 2 time points for the cinnamic aldehyde, and we see very similar rate constants over time. That's highly reproducible. And in both cases, of course, we run cinnamic aldehyde in every assay. And then also some words to the 2 assays which were developed in our lab. So for keratinosense, we updated the SOP in 2022. And cell distribution has also been standardized. Keratinosense is now routinely distributed by Accelerate. They sell this cell line without that you need a license agreement. You can just order them. They also offer assay-ready cells. But the standard cells, as they offer them, we have validated them in our lab that they are fully compliant by testing 6 chemicals, and we have a validation report on that. At the same time, some of you may have heard about the test guideline 497, which also looks at borderline cases, whether a chemical is borderline, and we generated an additional spreadsheet to evaluate the data and automatically detect what is a borderline case. So this is also part of the updated SOP. And now when you want to find the SOP, be careful, not just Google, because Google somehow finds old versions still around from the European Commission, but you have to go to the website of the DPL protocols, where really the latest protocols are posted, and there you will find the protocol, and you can also find the supporting files with the new spreadsheet. The same is also for the kinetic DPRA. We made an updated SOP in 2022. And what is here very important is that you don't have interference by fluorescence or absorbance by the test chemical, especially if you have complex chemicals. We have provided detailed guidance how you can check for interference in Annex 1 to the SOP, and we also added a special additional experiment where you have what we call an inverse substrate addition. That's all explained in this annex, and that needs one more control plate to be pipetted, and this will help you to find out whether there is interference. And again, we made a spreadsheet which helps you to automatically calculate that. Again, this is available from this from the official EQUAM website. You again find the supporting information, you find the Annex 1, and you find the spreadsheet where you directly can evaluate the data. So everything is publicly available, but be careful to get it from the right source. So this brings me then finally to the outlook. We will go on discussing how many tests we need from what I've shown you already now, which directly will lead us to how we can combine this POD determination with the 2 out of 3 assessment according test guideline 497. And then I will provide you an outlook of a new paper which we actually will submit this week, where we also look at using human data. And then we will discuss what it would mean to incorporate further tests. And finally, I will present from my side case studies on 2 cosmetic ingredients, and Argel will provide more case studies from his side. Here, just the acknowledgements. I did co-draft these publications and had a lot of very useful discussions with Frank Gerberick when preparing this work. And in my lab, it was mainly Roger and Tarantina Haupt who had been generating these huge databases and doing the validation work and developing the assays. And we worked together with BASF in the validation, and we had a lot of interaction in the OECD expert group. And that's where we live in this building, and that's the team we work with. Happy to answer your questions.
Thank you, Andreas. I'm going to go ahead and pull up my PowerPoint. All right. Thank you, Dr. Natsch, for your presentation. I look forward to your presentation on the 14th, wherein we can gain a better understanding of the PEC3 values and the publications. Just a reminder to everyone, because I saw a question in the chat, this webinar will be available on our IIVS YouTube channel and our LinkedIn, and we'll also send that out via the email that you used for registration today. At this time, we'll start the Q&A session. Remember to use the chat box to type in your question if you can think of any. For questions not answered at this time, we will do our best to answer and compile the questions and share questions and answers to all attendees via the email used for registration again. And I'll also put up those useful references that Andreas had in his presentation, just in case you wanted to take a screenshot and look more into those before the February 14th, part 2. So the first question that we got was from Raul, and they asked, why use DPRA for the point of departure calculation if KDPRA will need to be performed again?
Yeah, I think that is for me. Is my final slide in where I present what we have been done in the past year with our clients at IIVS, and I should say that pretty much I put that from the also regulatory perspective and from the testing perspective, because these are 2 different questions. One is to assess the hazard using the 2 out of 3. So when we have clients that have an unknown sample, they don't have any data, they can proceed to use the 2 out of 3 to determine if the sample will be positive or negative. If it is negative, the test will be end up there. But if it is positive and if they have interest on having the potency data, they will proceed to the KDPRA and perform the PC3 calculations.
Thank you, Argel. Our next question is from Stefan, and he asked, where we have chemicals with large under or over predictions, have you compared with other in vivo results such as MK and Buehler, if available?
Yes, I have looked at this data. The problem with the Buehler and the guinea pig maximization test is that we don't have real potency data, so we cannot directly say how potent they are. That's why when we had the data which are often really looked at is something known from the clinic, are these frequent sensitizers or are these rare sensitizers. So the discussion which we provided is mainly in relation to the human data.
Thank you, Andreas. The next question that we got was from Rule. How good were the equation 6 and equation 5 models as compared to equation 1 and 4?
Yes, so it is basically equation 5 is the most predictive, but it's only like 2 or 3% better in the regression coefficient. But the 1, 4, and 5 all integrating kinetic deep array are better than equation 6, which has only the 2 cell-based assays. So, which is in line with what we know of the adverse outcome pathway. Reactivity is the key initiating event, and for all chemicals together, it's needed for the best models. But also with the 2 cell-based assays, you can have a decent prediction, especially for those chemicals which are not reactive.
Thank you for the additional questions I'm seeing come in. Stephen asks, what do you think might be a realistic timescale for this approach, gaining widespread regulatory acceptance?
Yeah, that's a good question. On the one hand, at the moment, we don't have really official regulatory acceptance for any quantitative point of departure, because that is mainly used by industry to find a safe dose by either the cosmetic or the fragrance industry, and there's not really an OECD guideline for a point of departure. Also, the local lymph node assay was only validated for the GHS subclassification 1A and 1B, and for that, the kinetic deep array is now fully accepted at the OECD level. But there is a work ongoing at the OECD defined approach group to bring the point of departure also into the test guideline for quantification. But this discussion have just started. They will start it as a pilot with the SARA model, which has been developed by Unilever. At the moment, this approach is not in the pipeline there. I would need to find a country who's willing to submit it to the OECD. That would be the essential I would need. As an industry, you cannot propose something to the OECD. So, but if there's some country representative who wants to do that, I would be happy.
Our next question comes from Jens Venslow. Is there a reason why a point estimate for EC3 is used, although your regression model would also provide probability estimations?
Yeah, I mean, that's the question of a probabilistic versus a deterministic risk assessment today. What we do in QRA is a deterministic model where we have a defined point of departure, which leads us to a defined NESIL. And from that, we use uncertainty factors to use uncertainty. Of course, in a probabilistic risk assessment, we could use the uncertainty which we already know from the model and uncertainty from the in vitro data and translate that to a distribution, which then could enter into a probabilistic risk assessment. But that is not what's currently done. And so we really try to make that in a way that we can sort of plug into the QRA as used by industry today, because that was the key question when QRA was evaluated by the Joint Research center in Europe for its validity. They said, well, that works well, that looks good, or it's all logical, but you use an EC3 from the LLNA and you will not have this EC3 from the LLNA. What will you do? And that was really what we wanted. That was really the gap we wanted to fill here.
Thank you. Amelia asks, can this approach become part of the OECD test guideline? If so, will you pursue its inclusion at some point? Which I think adds to our previous question.
Yeah, I think I was thinking about submitting it. As I said, I didn't find yet somebody who had the money and the will to do it. I mean, it will need some preparation and some discussions before we are ready for that. But maybe we will try to put it on the OECD program starting next round, which is next autumn. So that's certainly possible. The benefit here is that everything is published. Everything is finalized, the spreadsheet is ready, the application is ready. So this will be a relatively easy implementation because everything is done. And we will now also publish the third paper on human data. So all the data analysis is done. Once we would propose it for the OECD, yeah, all the pre-work would have been finalized.
Good luck with that next fall. I don't see any additional questions in the chat, but I did have one for you. Are there any limitations on the type or class of chemicals that can be used for the EC3 prediction?
Yeah, I will go into some more detail on in the second webinar, but basically, as the kinetic deep array is quite important, chemicals which are under-predicted in the kinetic deep array are the ones which are under-predicted, as I pointed out. And there we know that we have especially these chemicals which have a specific reactivity with lysine residues. They will be under-predicted, but they can be recognized if you do the normal DPRA and you find that your chemical is reacting with lysine, but not with cysteine, which is a rare case. There are a few chemicals for which this is the case, but if you observe that in the DPRA, you will see that you then get a negative kinetic DPRA, and that should be treated with caution. That would be a case where you would go to equation 6, just using the cell-based assay, or you do then an expert assessment, taking this important reactivity with the lysine peptide into account, because we know that, and we published that lysine reactive chemicals on their own have a quite significant sensitization risk. And then you have, of course, things which are not applicable because they are fluorescent or they have an issue with the assay. But there's a workaround there. You can do a normal DPRA with HPLC, but run it in the kinetic mode. In principle, you can just make a normal DPRA and inject it into the HPLC over time, which you wouldn't want to do as a routine because it's quite tedious, but you can do it on single chemicals where you have an issue. So there's also a workaround there. And then the third question is, of course, the complex cosmetic ingredients, the mixtures, and I will go into that in the next webinar.
And I just wanted to add to Andreas' response that you need to think about from the perspective of the applicability domain of these assays, because any type of chemical that will not give you good results, probably it will not be great in this analysis because you need all those parameters to be input in the PEC3 calculations.
Thank you both. Cindy asks, can Andreas explain a bit more about the application of factors of 2 out of 3 to account for uncertainty? At what point in the risk assessment process are these used? For example, would you divide the PEC3 by 2? Is there good certainty?
Yes, that's exactly. If I have a chemical, let's say it's a Michael acceptor, which has a a beta substitution, and I get an EC3, a predicted EC3 of 5%. And then I look in my database to have other chemicals with similar structures. And I look at what is my in vivo data, what is my predicted value. And if that matches quite well, then I would say I'm in a domain where I can well predict. So I trust my value and I would divide my PC3 by 2. If I have a general chemical where I have no suspicious that it should be wrong because it's not out of the applicability domain, but I don't have very close analogs, we would start with a default factor of 3, because then we cover the 75% percentile. And then when I know there's something wrong, not wrong, but there's a reason why I think I'm out of the applicability domain, I have related chemicals which were not well predicted, then I would probably need a higher assessment factor, or I might need to go to further testing or other tests to further narrow down my uncertainty.
Thank you. At this time, we have no further questions. So thank you for joining. And remember to join the second part of the webinar next week. You've already registered if you registered for this webinar.
Thank you, everyone, and see you next week.
Thank you.
Thank you.
Bye.